Illumina for inviting me here today to talk to you about our work in pediatric cancer translational genomics. These are my disclaimers. So what motivates us in this effort? So although cancer in children is rare in this country, it remains the leading cause of disease-related death for patients. Excuse me. We are working together with the Dana-Farber Cancer Institute and Children's Hospital to profile pediatric cancer with ctDNA assays in the hopes that we might improve patient outcomes. And through my work at the Broad, I've actually had the opportunity to meet some of the patients impacted by these diseases. And that gives me a deep sense of motivation through that direct-to-patient research that I really want to make an impact and apply our tools at the Broad to this problem. So this effort is called BrightSeq, that we're trying to develop assays in our lab that will be offered in a distributed model with Children's Hospital. We are using whole genome sequencing and panel sequencing to profile these pediatric cancers. And the signal we're looking at today is mostly driven by genomics. So copy number events and fusion detection will allow us to quantify the presence of cancer in patients over time. And this can really help to power clinical trials in pediatric cancer, where they either want to stratify patients onto different treatment arms, and also to understand when patients are responding to therapy or developing resistance. But we recognize this really isn't the complete picture of the pediatric cancer genome. And so we were really excited when the opportunity arose to test out 5-base on these specimens and see if we can add the signal of differentially methylated genes and apply that to either identify tumors more sensitively that might not have these genomic events, or to subtype tumors. And it's really important, particularly in pediatric cancer, that we're able to generate this data from the same specimen, because when we're talking about children with cancer, we might be working with a very small amount of material, less than a tube of blood or a very small biopsy. So what have we done? We've generated 96 whole genome sequencing with 5-base chemistry. Some of those we've also profiled with our standard methods. And we've also used a targeted panel assay to more sensitively do deeper sequencing in 12 pan-cancer neuroblastoma cfDNA samples. And our goal here in this evaluation is really to assess this technology for application in this use case and potentially enable in the future molecular clinical assays. So in this workflow, we are pretty much using it out of the box. We're using 10 nanograms of input from cell-free DNA and 100 nanograms from FFPE. And from the extracted material all the way through to a prepared library, it's about 5 hours of effort. And as a large lab where we're running hundreds of thousands of samples, being able to use a workflow that's efficient and amenable to automation is really important. So this is really encouraging. It's another 2 to 3 days to do the sequencing and use the Dragon tools to do the analysis. It's been particularly helpful as the sort of analysis is already set up and we didn't have to build a bunch of new bioinformatics pipelines in our lab to look at the signal of epigenomics. The first thing we wanted to do was make sure our existing genomic analyses would work that we're using to profile these samples. And one of our major outputs is tumor fraction quantification from low-pass whole genome sequencing. And we wanted to test that alongside the 5-base workflow. Can we still quantify the samples using the same method? And you can see here the copy number profiles are concordant, and we have very accurate tumor fraction estimates between 5-base and our standard method. We also wanted to look at structural variants. Pediatric cancers are often driven by fusion events that we need to detect and quantify over time. And so here I'm showing the EWSR1 Fly1 fusion, which occurs in Ewing sarcoma. And this is really encouraging, especially for this region, because it happens to be GC enriched. And other methylation sequencing technologies, it's really difficult to align these reads and call these events. Small variant detection also looks good. We compared this to our standard usual benchmarking. For SNPs, if you want to see more of this, I have a poster later. And for somatic variant detection, we're using the targeted panel so that we can see the variants at low tumor fraction. And we were encouraged to see that the expected genes that we expect to be mutated in neuroblastoma, we're calling the right variants. Just to show you what the data looks like itself, this is a 20% variant in ATM from that data set. And you can see the signal of methylation at the orange arrows on the right. So now I'm going to switch gears. So given we were able to generate the genomics data for this effort, we then were released to sequence the rest of these very rare and valuable specimens. So we are looking at pediatric renal cancer here as a use case. There are two kinds of renal cancer in children. 80% of the cases will be Wilms tumor, which is the more prevalent histology, and it tends to be less aggressive. Identifying the MRTK samples is important so that the patients can get the right treatment. Often surgery and biopsy is not possible in these patients. Biopsies are actually not safe, similar to the last study. When patients don't know which cancer they have, because they're not going to get surgery, they will often get treated with the prevalent histology treatment. So they're not going to get the right drugs. They won't respond. And so really what we want to do is identify these tumors earlier. These different cancers have different events. Wilms tumor is often, but not always detectable using our standard conventional whole genome assay. But MRTK is much quieter and often most of the time will not have any copy number events. And just to put numbers around this, we only really detect about 20% of the MRTK cases when we're using our standard methods. So this is what we're trying to improve. So our plan is to use FFPE against cell-free DNA from healthy donors to identify features that are differentially methylated regions in these samples and train machine learning classifiers using the methylation signal so that we can better identify MRTK tumors and kidney cancer population. We have been working on this for a while with our collaborators at the Dana-Farber, and so we already had DMR models prepared. So the first thing we did was apply 5 base sequenced cell-free DNA samples from kidney patients to those models. And as you can see, and we expect for the Wilms cases, we are detecting most of the samples with the new methylation classifier, but those are also detectable by our standard genomics method. For MRTK cancer, we're actually identifying additional samples now that were not identifiable before with our standard method. And it seems like small numbers, but for pediatric cancer, which are rare cancers, actually identifying these is remarkable. And out of the box, just applying this data to our models was also encouraging that it was working. So our future plan is to now develop 5 base specific machine learning classifiers using differentially methylated regions that we're sequencing now in the lab. So for Wilms tumor, we're already partway along with that. We have about 3,400 DMRs for Wilms, and we have 700 for rhabdoid tumor. And the additional data that we're sequencing will be fed into these new classifiers. So our impression of 5 base is like this is a really strong addition to our portfolio for doing translational research in pediatric cancer. The process was very easy to run. The data was very high quality, and it's already adding to our knowledge here in pediatric cancer.